Welcome again to this session, another session of emergent topics in biomolecular magnetic resonance, organized by Lauren Andreas, Stefan Gugler, Christian Griesinger, Mei Hong, R. Palmer, Marcus Spexeter, and myself. And the idea is to present frontier research in magnetic resonance in biological systems. Today we have another very exciting session with Tatyana Polenova and Marcellus Ubbink. And please, these conferences are actually sponsored by ICMR-ES and Euromar. Remember that you are not supposed to record the lectures because they will be taped for you and then distributed through the ICMR-ES YouTube channel. Should you have any question, please use the Q&A section of the Zoom software, and then we will actually ask for you, or in the more informal session, you can also be able to participate in the meeting. That said, I think it is probably time to introduce the first speaker, and for that, I just leave the word to Professor Angela Cronenberg.
Hello, everybody, and it gives me enormous pleasure to introduce Tatyana Polenova. She's well known in the NMR community. Somebody once described her to me as a dynamo, and I've probably never met anybody who can pack more words in 1 millisecond than Tatyana can. And for those who don't know her, a little bit about her background. She got her undergraduate education at Moscow State University and then moved to New York to Columbia University, where she did her PhD in Anne McDermott's lab. She spent a short time at City University of New York at Hunter College, and then moved to the University of Delaware, where she is now, and where she rose through the ranks from assistant professor to professor, and she's been a professor there, a full professor, for the last 10 years. As you may be aware, and for those who are not, congratulations, Tatyana. She is the editor-in-chief of the Journal of Magnetic Resonance as of this year. She also serves as an associate editor for the Journal of Biomolecular NMR and on the Journal of Structural Biology. She is a really good citizen as it comes to helping the community, both the chemical community as well as the NMR community. Let me just remind you that she was very instrumental in organizing the High Field NMR initiative in the US and really pushed the discussion and conversation about this forward at all levels with funding agencies and among ourselves. She has organized numerous conferences. She was on the ENC executive committee. The Telluride workshops are sort of a gem amongst some of the small conferences. And personally, I have the good fortune that she's been a collaborator of mine for the last decades, and I've learned enormous from her. And I'm sort of a believer and proponent of solid state NMR, and sort of have one leg now in the solid state NMR. And with this, I won't take any more time from her, despite the fact that she will still finish her talk in time and speak very fast, and hand it over to Tatyana now. Please go ahead.
Thank you so much, Angela. This was supposed to be a surprise, and it was, and it still is. So I'm very grateful for you introducing me. And I'm also very grateful to be here in this forum. Actually, thank you to the organizers for this very nice opportunity and for keeping the discussion going over all of these months of this difficult year. And I hope this format continues actually past COVID, and even when we all can see each other in person again. So I am also grateful to Angela for introducing me to the world of HIV structural biology. And what I want to tell you about is our journey today if I can, yes, to the screen. So our journey and lessons learned from and about HIV-1 virus, namely how to peer into structures and dynamics of large assemblies with atomic resolution using magic angle spinning methods and integrating with other structural techniques. So before I start, this is a long-standing collaboration with Angela over a decade or so, and Pittsburgh Center for HIV Protein Interactions. At the University of Delaware, this is a work of a tremendous team of graduate students, current and former, and postdocs. I will be talking about today mostly the work of Man Lu, who's now a postdoc at the University of Pittsburgh with Angela, the current graduate student Ryan Russell, former postdoc Kate Quinn, Yun Han, Chris Sutra, Guangjin Hu, Huilan Zhang, and more recently Sucharita Sarkar, Roman Zadorozhny, Maple Wong, and Changmiao Gu and Brent Mungi. For this work, we have been fortunate to have help from Charles Schwitters at NIH in some aspects of the structure calculation, as well as long-standing collaborator on the molecular dynamics, my colleague Juan Perilla, and his student Alex Pryor. And also, we have been working very productively with scientists from Bruker, Joachim Stuppe, Ivan Sergeyev, the late Melanie Rosé, as well as Leah Hassan, Barbara Perron, and Rainer Kurmeli. So I will be mentioning people throughout this talk. Now let's first look into the HIV virus. As you may know, this is a causative agent of HIV/AIDS. And at the moment, there are more than 35 million deaths caused by the virus. It's a global pandemic, and over 38 million people are living with HIV at the moment. And despite the fact that there has been a lot of research into the biology and virology of the virus, there is no cure. So even to date, we are looking for new therapies. And of course, this requires understanding the structural biology and atomic resolution picture into the structures of the virus. And so the virus itself is 9 genes, which encode for 16 proteins. Today, we will be looking at the GAG polyprotein encoded by GAG gene and its products, matrix, capsid, and nucleocapsid proteins, which are structural proteins of the virus. The viruses, mature ones, look like this, shown here in the transmission electron micrographs. And you can see the conical types of structures, which are mature capsids, which enclose viral RNA, 2 copies of it, and a small complement of proteins for replication. So these conical capsids are built by about 1,500 copies of this fairly small CA monomer protein, 231 amino acid residues, mostly alpha helical, 2 independently folded domains joined by a flexible linker. And an important structural element of this protein is a cyclophilin A binding loop. That's the site of binding cyclophilin A and several other host factors. To build these kinds of conical structures, one requires about 216 hexamers, and the stoichiometry is varied, coming together with exactly 12 pentamers to form these kinds of curved morphologies. Okay, so when we started this project back in 2008, there was very little known about the organization of these capsids at the atomic resolution. And over the past decade, we now have atomic resolution models and atomic resolution structures. And I will tell you what we were able to learn from this. And so in the context of viral replication cycle, we're interested in the steps which lead to the capsid assembly, and also capsid uncoating premature disassembly, which is facilitated by the restriction factors. On the other end of the virus life cycle is maturation. This is a process by which GAG polyprotein is cleaved into its individual domains. And there is a lot of interest now in designing inhibitors that block this maturation step. So this is a new line of thought in the field of HIV. So how do we study these kinds of assemblies by magic angle spinning? So far, most of the work we have done included recombinant assemblies, and we have been able to work with GAG virus-like particles where GAG is uncleaved. So these are spherical types of assemblies, as well as with mature types of lattices, which are forming cones, as well as tubular assemblies. This is the subject of most of my talk today. And this tubular assemblies actually recapitulate the hexagonal lattice formed by cones. So there is a lot of structural work done on those to understand the organization of the capsid. We have also looked over the years into complexes of these capsid tubes with various host and restriction factors, as well as the maturation intermediates. And you can see that all of these structures are morphologically distinct, so even tubes have different types of morphologies, and all of these actually give rise to very high resolution magic angle spinning spectra. I'm just showing here a few of those to impress on you the fact that if you can make the samples correctly and nicely, you will get high resolution spectra. But more importantly, from this, you can immediately get information about whether the lattice is immature or mature based on distinct chemical shifts. Okay, so now, despite the fact that the spectra are such high resolution, turns out that these are quite challenging systems to work with. First of all, they are complex architecturally, so the viral capsids are pleomorphic, and pleomorphism comes in different stoichiometries of the CA units forming the capsids, as well as in slightly different shapes of these capsids. Okay, so we're interested in understanding what gives rise to that. There are also sequence variants which give rise to different NMR spectra, of course. These are highly dynamic systems, and I will show some data where you will see that the motions, and these are functionally important motions, so we need to study this not only to account for dynamics when we look at structures, but also to understand the function of the virus. And of course, we're interested in looking at functional interactions with human host proteins. So we're talking about looking at large multi-component complexes. And of course, this presents challenges in terms of the resolution and sensitivity. So we had to put a lot of thoughts on devising different kinds of samples, using different types of isotopic labels, and exploiting some model assemblies through mutagenesis, which I will talk a little bit about today. We had to think about how we do the experiments, conventional heteronuclear detection versus fast magic angle spinning utilizing proton detection. More recently, we also turned into dynamic nuclear polarization, where we can get some sensitivity enhancements and be able to capture minor species. And in all this work, it's essential to couple experiments with computation at the level of both molecular dynamics and quantum mechanics. All right, so let's first take a look at the tubular assemblies of CA. So as I already told you, this gives high resolution spectra. Since the protein is fairly large, it took us a few years to do the assignments. Yun Han and Man Lu, mostly it's work from them, they were able to assign 96% of resonances in different sequence variants, and this paved the way for atomic resolution structure determination of these assemblies. So Man prepared a series of samples, such as 1,6 and 2-carbon-13 labeled glucose, and acquired a series of carbon-carbon correlation spectra shown here with different mixing times, core data sets. And you can see that there are a lot of non-trivial distance correlations corresponding to distance restraints. When we take all of the samples and she put all of the restraints information together, we have over 1,300 of those, both ambiguous and unambiguous, which in principle should be a fairly large number to be able to get structures calculated. So she and Ryan Russell in the lab performed these calculations, and what they discovered early on is that one can get only structures of the individual domains and C-terminal domains based on the magic angle spinning NMR restraints alone. For those of you who work with multi-domain proteins, this probably doesn't come as a surprise, because typically it's very difficult to define relative domain orientations based on the NMR restraints alone. These are fairly short distances. And there are a couple of ways we chose to tackle this problem. Today I will talk about integration of magic angle spinning with low resolution cryo-electron microscopy maps. And what Ryan has done with Charles' help is develop the procedure for joint refinement of the magic angle spinning NMR parameters and the cryo-EM distance maps. And he was able to get structures of the hexamer unit of the tubes. Okay, so just to give you a little bit of statistics, these are very nice, very tight structures, 1 angstrom heavy atom RMSD, but these are still not the structures of the tube, right? So how do we get the structures of the tube? Here's where molecular dynamics simulations became very important. Alex Pryor, a graduate student in Juan Peria's lab, took the structure of the hexamer and the NMR restraints, and he built the structure of the tubes using molecular dynamics simulations. So now we have this detailed atomic resolution structure of the capsid tubes. Let's look and have a glance of the structural features. So first, let's look at the interfaces, which are important in holding the structure together. There are two types. One is this dimer interface here, as I'm pointing with the pointer. And this interface is formed by residues of helix 9, as shown here. They're hydrophobic interactions from these 5 types of residues. And what we found is that the interfaces in the tube are conformationally heterogeneous. This is also consistent with the experimental NMR data, of course. And so not only that, but statistical analysis identified 9 distinct dimer interface clusters. Surprisingly, in all of them, tryptophan 184, that's a critical interface in the dimer formation, as shown by the work of Angela in solution NMR. This tryptophan is in a single rotameric state, and there are direct hydrophobic contacts between the neighboring indole rings. So there are also some interesting water-mediated contacts with the side chains. Another type of interface which is important is this trimer interface shown here. And in fact, there are 2 types of trimer interfaces, which are shown in orange and in blue in those pictures. These are formed by residues of helix 10 and 11, and you can see that the hydrophobic core is formed by 7 of those residues, and it's supplemented by polar interactions from K203 and E213. There are 2 equally populated clusters. What's important is we learned from the intermolecular distances, such as C-alpha distances from leucine 205, that these are similar to the distance distributions from cryo-electron tomography structures of intact viral capsids, and that's different from what you see in the crystal structure. So the structure of these tubes is a more relevant structure biologically. Another thing which is coming very pronouncedly in the NMR structures is the conformations of the functionally important regions. One of those is flexible linker, which connects N and C terminal domain. And you can see that there are 4 conformational clusters here. This is also consistent with the conformational flexibility of this linker, which positions the 2 domains relative to one another. A cyclophilin binding loop, which we found to be quite mobile based on our dynamic studies, indeed adopts 4 distinct clusters with different populations. Okay, so again, that's consistent with the fact that it's dynamic, and beta hairpin adopts a single conformation. And so side chain orientations are very well defined in the structure, unlike cryo-electron microscopy structures. And this is where NMR shines, of course. We have a lot of carbon-carbon distance restraints which define the relative side chain orientations, and shown here are just representatives for the 9 of them, pretty tight ensembles. Another important thing that we learned from the structure is that it's very different from the structure of the flat hexamers. This is an X-ray structure, and you can see that tubes, of course, they have curvature, and this is a superposition between the crystal structure. Here, the beta hairpin is in green, and the NMR structure, the beta hairpin is in yellow. Okay, so there are some very distinct conformational heterogeneities which are present in the NMR structure based on the chemical shifts, and then they're borne out in the calculations as well, such as this residue R18, which is critical for the pore formation in the NTD. And in the crystal structure, this is a fully conformationally homogeneous type of side chains. All right, so there are details which are very different from the crystal structure, which shine light on the more biologically relevant conformer. So in this work, we were able to get atomic resolution structure of the tubes. We had to combine magic angle spinning NMR restraints with information from the low resolution cryo-electron microscopy maps. And what's important, we were able to both capture and quantify the conformational heterogeneity in the structure, which appears to be a hallmark of CA assemblies and defining the pleomorphism. Another lesson that we learned over the years is the fact that the capsid is dynamic. And let me see if I can, okay, so if I can start this movie, this is actually an MD trajectory determined by Juan Piri, calculated by Juan Piri and late Klaus Schulten. And this illustrates the types of motions that occur in the capsid. Capsid is not a rigid structure at all. There are a lot of motions, which you can see here based on the MD trajectories. There's motions, breathing motions of individual hexamers with respect to each other. These are motions of the cyclophilin A loop shown here in red, You will see that when this is zoomed in, and motions of the flexible linker. So over the years, we were able to measure and quantify these motions and connect them to the function of the capsid. So I will not go over the studies. This is all published work, and you can look it up if you're interested. But I want to tell you about a more recent study. We were intrigued by the discovery, again, from molecular dynamics simulations made by Juan and Klaus, about the fact that they see a lot of different kinds of coupled and correlated motions which span multiple time scales, and they appear to be inherent feature of these conical capsids. And what's interesting is, as shown here, pentamers are in green. They appear to be much more rigid than the rest of the capsid structure. So they concluded that the capsid's acoustic mechanical properties are an inherent feature of the system and linked to its stability. Of course, this is a simulation. Can we probe anything like that experimentally? In particular, can we see differences between the motions of the pentamers and the hexamers in the capsid structure? And it's very difficult for one very simple reason. Remember, there are only 12 pentamers in the capsid, so sensitivity-wise in NMR experiments, it's very limiting. So this is when we turned attention to the model building block for the capsid, and these mutants, which were devised by Owen Pornelius and Barbie Ganser-Pornelius, is a quadruple mutants where two key residues of N-terminal domain, N21 and A22, are mutated to cysteines. If you do that, and make cross-linked constructs, you form pentamers exclusively. There is another mutation, A14, E45C, where the cross-linked construct forms hexamers exclusively. So we exploited these types of mutants to be able to get a glimpse specifically into hexamers and pentamers. And what we've done is we've prepared 3 types of assemblies. One is the tubular assemblies, which I already told you about. So this is containing carbon-13 and nitrogen-15 labels on all the atoms. And we're looking at the, of course, hexamers alone. And we also prepared cone-like assemblies of A204C mutant, cross-linked. It forms these very nice conical structures. And when the entire protein is labeled, we will again be looking at hexamers predominantly in the context of those cones, not tubes now. And another construct that we have looked at is where we labeled these cross-linked pentamers with carbons and nitrogens and prepared mixed cones as shown here. So we will be only looking at pentamers in the context of these cones. And so all of this give very nice spectra shown here. These are just, snapshots of the core data set, so we can get a lot of information. And lo and behold, if we do different experiments to probe the motions, for example, NC-REDOR, which gives us information about millisecond timescale dynamics, this experiment tells us that, yes, indeed, pentamers have very distinct motional signatures from hexamers in tubes or cones, which look more similar. And this is only true about slow motional scales, as you might expect. Right, so this was a very interesting sort of initial discovery, but we ran into bottlenecks. So all of this are very insensitive experiments. So you can imagine that with this kind of mixed assembly, we need many hours of experiment time, and indeed 5 days for a single 2D. So it means that we can't get more information at this point. And this also brings me to the next topic, the fact that we have roadblocks, sensitivity, not only in the HIV-1 assemblies, but also in looking at a larger biological assemblies. So of course, first of those is the need for isotopic labeling. So we cannot work with, in situ structures. At least we need to do something clever for that. And then we work with large molecular weight assemblies, multi-component minor species, dilute assemblies, and all of them are disordered or dynamic systems. So in addition to HIV-1 capsid assemblies, this problem is inherent in other types of systems we worked with in our lab, such as microtubule and actin-associated protein assemblies. And more recently, we started getting interested in looking at natural abundance small molecule inhibitors bound to proteins. And so how can we tackle these issues? Over the past few years, we looked at several approaches. starting from fast magic angle spinning frequency and proton detection. Beautiful technology, but I can tell you that unfortunately, at least for our systems, it doesn't work well in dilute samples, so for minor species detection. Another approach is to use PAC, paramagnetically assisted condensed data collection, and exploit the short relaxation time, but this requires introduction of paramagnetic dopants into the system, not compatible with all sample preparations. Non-uniform sampling is a technique which I would strongly advocate for. It doesn't have any inherent drawbacks, and it actually gives us true sensitivity enhancements. The only problem, it requires overcoming some potential energy barriers. So I have to convince my students who come to the lab that they need to spend some time learning how to do these experiments and process the data. And if we combine non-uniform sampling with PAC, has same drawbacks as PAC. Dynamic nuclear polarization is also great, but at the moment, I think many of us are trying to resolve the resolution problems at low temperatures. And more recently, we have turned attention to CP-mass cryoprobe, which I want to spend the last couple of minutes telling you about, and we did not see any drawbacks for that. So this is a technology which was developed by a team of Aliyah Hassan at Bruker, and we were just very fortunate to be included in this work. So in fact, Angela, Kate Quinn, Joachim Strupe, and Ivan Sergeyev went and did a couple of trips to Switzerland to be able to use this cryoprobe, and we also collaborated with Chris Jeroniak and Antoine Loquet on combining the results from different labs to see what the advantages and limitations are. So and basically what we saw is that there are significant 3- to 4-fold sensitivity gains obtained in these experiments, which means that we can reduce the experiment time by 10 to 16-fold. And we were able to demonstrate the benefits on 5 different biological assemblies. For this discussion, I will just present the data on 2. One of them is the conical assembly, where the CA is uniformly labeled. You can see those cones here. And this is a benchmark sample for sensitivity. And another sample is a tubular assembly, where we have only nitrogen-15 labels. And this is a sample we were particularly curious about the possibilities of doing natural abundance carbon-13 spectroscopy. We were very pleased to see that one can get very high sensitivity data sets using 2-scan cord with a signal to noise of 115 roughly, and everything looked very nicely. So for this type of sample, we were able to reduce the experiment time significantly. So basically, it took only 4 days to collect the basic experiments, 2D cord and 3D and CACX and COCX for assignments. And you can see the quality of the data set here. We were able to assign the resonances in these conical assemblies using those data sets alone. And perhaps the most exciting result is on this natural abundance carbon-13 sample, where we could get 2-dimensional NC correlation spectra, NCO and NCA. And you can see that the resolution is very nice. But for me, also a very exciting result was that we were able to get nitrogen-nitrogen correlation spectra, which you typically don't do in the samples because of low sensitivity. With the cryoprobe, you can see that we can get this very nice data set. And in fact, there is a lot of information which is complementary to what we can get from the nitrogen-carbon correlation spectrum. Okay, so I want to stop here and just conclude by saying that I see a lot of opportunities for magnetic resonance and structural biology. We can provide information which none of the other techniques can. So magnetic resonance can inform us on atomic level detail and dynamic systems which are invisible by other techniques, as well as tell us about chemical transformations in biological systems. And to do that, it's also beneficial to integrate multiple approaches. We still need to overcome sensitivity and resolution roadblocks. And if we overcome those, we will be able, hopefully, to work in the context of intact viruses and cells. I would be very interested to see if we can do natural abundance NMR in these kinds of systems. And just a concluding slide, essential ingredients are all kinds of experimental techniques, magic angle spinning, NMR and dynamic nuclear polarization, computation, and integration with other methods. And I will stop here, put up an acknowledgement slide once again, and thank you very much for your attention. sharing the screen.
Angela, are you taking care of the questions?
I can. Okay, the first question is from Ralph Masevsky. Are the flexible regions in the capsid structure more or less likely to mutate in nature?
I don't know about more or less likely, but there are mutations in the cyclophilin A loop, which are so-called escape mutations. What they do is they abolish the dependency of the capsid on cyclophilin A. So yes, there are known mutations in the loop which are functionally relevant. As far as the frequency of the mutations, I must confess I have not looked, so I don't know if it's more or less likely.
Could I ask a question while the others are arriving?
Sure.
You mentioned this interesting interface with tryptophan 184, which looked like it was next to a valine, perhaps. Did you check, or did you find if there were interesting ring current shifts or characteristic ring current shifts in the protons of those nearby methyl groups?
Oh, it's an excellent question. We don't have the proton data yet, so we do, but we didn't look carefully. But that's, thank you for asking. Yes, on carbons we don't see anything unusual.
Christian, go ahead.
In some of the structural pictures that you showed, there were little holes. I mean, most of the time they were with hydrophobic amino acids. Is something going in and out, or is this really a tight packing such that these little pores or whatever that were indicated would always be closed?
So there are, and let me see if I can put that slide back. There are channels, the pores in the structure. So if we go back to the structure of the hexamer, this is the spore here, and people think that there are nucleotides going in. Can you see the screen share? Yeah. And obviously they're penetrable to water as well. So,
but it's just water and maybe ions or so to
water, ions, and nucleotides.
Mm-hmm. Okay.
Any other questions? I don't see any more in the chat, so maybe anybody on the panel wants to ask some questions.
I just had a curiosity. You said that cyclophilin was actually dynamic, and then you related that to the dynamics in solution. But are we talking about the same timescale and the same kind of motions, or just the fact that they are actually embedded in this superstructure actually creates a different kind of concerted motion? I think you mentioned partially that, but.
So if we look at the individual molecules in solution and in the assembled structure, the structural elements dynamics is quite similar, I would say, for example, cyclophilin A loop. And it's surprising that in the assembled structure, the cyclophilin A loop is very mobile. As far as the concerted motions, you need to have the assembly to have those. And
those concerted motions do affect cyclophilin as well.
So cyclophilin A loop motion, I would think that it's independent of the longer time scale concerted motions.
Okay, thank you.
There is another question from Grace Rayappa. Can you comment on where the current inhibitors bind?
So they bind at inhibitors of the maturation. So they bind at this SP1, and I see if I can get the structure for you here. Sorry, jumping around in the slides. So the maturation inhibitors bind at this SP1 region here, and it forms a 6-helix bundle in the immature variants, and they go into the pore. So we have structures now of the inhibitors bound there, not published yet. So I didn't talk about it today, but it's pretty well known that they go into the SP1 tail region.
And Lucas Simons asked, do the dynamics in the hexamers and pentamers depend on the local curvature of the assembly? in the tube or the cone?
So for hexamers, we don't see much difference, as I've shown you for the tubes and the H04C based cones. The dynamics are very, very similar. So pentamers is a standalone entity in the cones. They're not present in the tubes. And they appear to be more rigid. But we have, unfortunately, very limited sets of data at this point because of sensitivity limitations.
And no more questions in the chat, or as we say, no more questions from the chapel when it was the ENC.
I think we're also pretty much in time, so we probably could just go to, I mean, we have this informal session at the end where hopefully everybody can stick on, and then more questions may arise, and I guess we just go over to the next talk. And Gerard Cantas was so nice to agree to introduce Marcellus.
Thank you very much. Do you hear me? Okay.
Yes.
It's a pleasure for me to introduce Marcellus Ubbink. Just a bit of history. He studied in Utrecht and in Leiden, biology. Got his master's in 89, cum laude, which is only happening for a few % of the students. Then he moved to Leiden to start his PhD research in the metalloprotein group, where he worked on the redox chain, methylamine dehydrogenase redox chain of Thiobacillus faucitus. Marcellus was a fantastic student for me. He was the only student who was able to solve any problem that I gave him. And for a supervisor, that is marvelous. After a couple of years, he got his PhD again, cum laude, in 1994, and then he moved to Cambridge to Derek Bendel and John Gray to study there again on NMR of proteins, partly on cytochromes, and was very successful after his postdoc period in Cambridge in 1997. He got offers from many sides, so it was a bit difficult for him to choose, but we finally were able to make him an offer from Leiden that he couldn't resist. So he moved to Leiden to become an assistant professor, and then he moved through the ranks to become a full professor in 2010, 10 years ago now, full professor of biochemistry. He became famous for his work on paramagnetic proteins. He devised several techniques to use the paramagnetism, the natural paramagnetism of some proteins, but you can also attach paramagnetic probes to have artificial paramagnetism. And it turned out that it can be a very strong tool to assist in structure determination and also in the determination or the study of protein reaction mechanisms, enzyme reaction mechanisms. He's presently very strongly interested in biological electron transfer, in protein structure determinations, in protein mobility, the application of magnetic resonance techniques, not only NMR, to the study of proteins. And he's very successful at that. He has a lot of students, big group. And I'm very curious to hear today what he is going to tell us. He's locking up metals. It's a bit as the Americans 4 years ago, they shouted, lock her up, lock her up. But Marcellus is doing this with metals. So he shouts, lock them up. But that seems to be very fortunate for his techniques. So I would be happy to give the floor to Marcellus, please.
Well, thank you very much, Gerard. This is a really nice surprise to see you here in this audience. And thank you very much for your kind words. I'm blushing a bit here, but I hope you cannot see much of that here. Also, I'd like to thank the organizers for having this great initiative. I think it's very useful. We also save a lot on CO2 emissions in this way, which is good. And I've really been enjoying the talks so far, and I'm happy to talk a bit here about paramagnetic NMR. As Gerard said, I'm advertising this technique already for quite a while, and I'll also do that today. So I'll start sharing the screen. All right, I think you can see it now, right? So I'll introduce a little bit the idea of paramagnetism in NMR, because not everybody will be familiar with it. I think at least to the extent that we'll use it here in this talk. So I just indicated here a protein with a nucleus inside, and we have an external field like we will do in NMR. We'll throw some radio wave irradiation at it and then get some signal out. That is what we will do in NMR. And then for paramagnetic NMR, we're simply going to add an extra bit here, a paramagnetic probe, a metal in a cage. That is why we lock them up. We need to somehow attach that to the protein. And then what can we get? Well, this will have an effect on the nucleus because of the interactions, for example, the dipolar interactions between the unpaired electron and the nuclear spin. So we'll get a shift, for example, like here, the pseudo contact shift is the most relevant one in this case. So a shift of the peak position. We also get line broadening called paramagnetic relaxation enhancement. Already Tatyana referred to it for solid state NMR briefly. So we can use that. And what also interesting feature of these paramagnetic tools is that we can use it to create RDCs, so residual dipolar couplings. Right, normally you have a J coupling, and then if we add the probe to the protein, because of the interaction that the probe has with the external magnetic field, that the effect is so strong that the molecule will slightly align in solution, just like adding gels or bicels, for example. But now the alignment is caused by this metal here, and that will cause a residual dipolar coupling that you can measure just like normal RDCs, but now paramagnetically induced. That has advantages because, of course, the alignment is then automatically inherent in the whole protein, and it is distance independent. So you can measure these three things, pseudo contact shifts, paramagnetic relaxation enhancements, and residual dipolar couplings. And just briefly to talk a bit about what you can use this for, I'll give a little bit older work, just as an example of what you can achieve in structure calculations, and then come to a few new probes that we have been developing recently. So this paramagnetism provides you long distance information, right? PRE is roughly up to about 35 angstrom or so, depends a bit on the size of your molecule. PCS can be at least up to 70 angstrom away from the metal, so quite far compared to a normal NOE, for example. You can study lowly populated states. PREs are especially sensitive to that. So dynamics in protein-protein complexes, for example, and I mentioned already this alignment here. But you need this paramagnetic center. So it needs to come from somewhere. Sometimes you can use metals that are already present in the protein. That could be iron, copper, manganese, but that rarely is suitable. So often you need to introduce your metals, and that can be by substitution, for example, of calcium with lanthanoids, or copper with cobalt. Copper itself is also paramagnetic, of course. Sometimes cobalt is more useful. You can also use stable radicals, spin labels. The well-known MTSL, for example, has been used for a long time in EPR already. You can also use that in NMR, or metals in a cage, nickel, cobalt, manganese, lanthanoids. And I'll be especially talking about the lanthanoids because they are most favorable for this purpose. Just to show that indeed you can get 70 angstrom or 7 nanometers in a PCS. Here you see an example of that in a complex that I'll be talking about soon, of a particular proton that was here in the partner protein, and the metal was all the way on the other side. This is 7 nanometers, and this is the pseudo contact shift that you can get from that. So indeed you can measure over long range distances. So as I said, we use lanthanoid probes in preference. Ideally, we attach them via 2 arms. There are many probes by many groups developed now, and there's also plenty of reviews giving all these different probes. They all have advantages and disadvantages. And we like to stick with the 2-armed probes here that we attach via 2 cysteines to the protein. The lanthanoid is in a cage and attached via 2 sulfurs then to form disulfide bridges. And you see a model of that on the right-hand side here. They have advantages. They're quite rigid relative to the protein, so you get large pseudo-contact shifts because of these 2 arms. They are tunable because you can put it in different lanthanoids with different paramagnetic strength. You can get strong PREs if you put in gadolinium, which is a strong PRE agent as a lanthanoid. They can create this alignment for RDCs. And as I said, there's many variants available now, many different types of probes. But of course, these probes need to be synthesized. That's not always trivial. Labeling can be more difficult than a simple MTSL. You have a bigger, bulkier group. In this case, you need 2 cysteines on your protein, like for example in an alpha helix here. And it sometimes is charged, so that might influence protein-protein interactions, for example. So you need to always consider also the drawbacks. So 2 of the probes that we use a lot are the so-called clamp 5 and clamp 7. Clamp is caged lanthanide NMR probe, or lanthanoid these days, officially in the nomenclature, I think it's called, developed by Peter Keizers and Wei-Min Liu quite a while ago now. But still, these clamp 5 especially, we made large quantities of it, relatively many grams, and that is used still regularly. And so you see here that they have a cyclin ring here in which the lanthanoid is maintained by these 4 nitrogens, and then you have 4 oxygens that come in, as you can see here, from in this case the bottom, to form a linker. And in this case, the oxygen is coming from a pyridine, oxide pyridine here, and on this side as well, and here 2 amide groups. This one is a similar one, but has these nitrophenol groups here instead, and has a different charge for that reason. And so this is 3+ and this is 1+, but otherwise it is rather similar. So here you just see an example of what you can get, the types of pseudo-contact shifts with the clamp 5 here loaded with the ytterbium in this case. So a moderate paramagnetic center of moderate strength. And you see here always the diamagnetic control spectrum, and in red here the paramagnetic sample. So you always make 2 samples, one with the control lutetium, in this case is diamagnetic, and the ytterbium is the paramagnetic, and everything else is the same. So you can read off these typical pseudo-contact shifts that are all on a slope of a line with a slope of 1. Yeah, so on the ppm scale, that is. And that has to do with the fact that the nitrogen and the proton, or the carbon and the proton, are very close together relative to the paramagnetic center, so they have more or less the same pseudo-contact shifts. And then that you get these typical patterns here of lines, either negative or positive shifts. So they're very easy to measure and very easy to you can accurately get this shift. Now, as I said, I'll just give one example of what you can do. This is structure determination of this particular complex. And Gerard mentioned already, my interest has been for a long time, and that is thanks to Gerard, the redox proteins. So we have here cytochrome P450 with a heme group sitting in the middle with an iron there. And this is a partner protein, ferredoxin, putidorodoxin with an iron sulfur cluster that needs to transfer an electron from this iron sulfur cluster to this heme group. And we were interested to see, can we determine how they bind together in solution? At the time, there was no crystal structure, so we thought maybe we can solve this with paramagnetic NMR. So the idea was that we take the proteins, we put probes in different positions, of course not all at the same time, but one at a time, either here or there on the other protein, and then create restraints, so paramagnetic effects from one probe onto the other protein, and use that to dock the 2 together. And this was work by Yoshitaka Hiruma, a PhD student in the lab, and Matthias Haas, a postdoc, who worked nicely together to solve this problem. So here you see an example of the type of data that you get then. We take the PREs, in that case we use gadolinium here to get the strong PRE effects, and you see The red peaks are much weaker in some cases, and that means that they're broadened out because of the PRE effects compared to the lutetium here, the blue ones underneath, that are diamagnetic and come up. The same here for the pseudo-contact shift. Now we use thulium, a really strong paramagnetic center, and you see these pseudo-contact shifts from one protein onto the other. And here an example of measuring these RDCs. That is not so trivial. It's hard to get these because the signal to noise was quite low. It's a large complex of more than 60 or about 60 kilodaltons. And we measured there an HSQC and then the Trosi form, and from the difference in the distance there, we can then get the diamagnetic RDC and the paramagnetic one, and from that, the diamagnetic coupling and the paramagnetic coupling, and derive the RDC from it. So to make a long story short, we collected a lot of data with different probes in different positions, and then acquired all these different restraints here for the docking of the two proteins together. This was a rigid body docking, so not very complicated. It was overdetermined, of course, with all these restraints. But I do emphasize that you need these different methods. So the PREs are really sensitive to minor states. The PCS are not unique in the distance because of these tensor orientations that are, they define a center of distances where the nucleus can be, and not one particular position in space. So you need multiple probes and you need multiple types of restraints to do this reliably, because each of these have their own limitations. So collecting all these then resulted in this structure here. So here you see the P450, the cytochrome P450, and here the putidaridoxin. And these are the 10 best solutions that you get when you dock it many times, like you do with NME structures, and you do multiple calculations. So the RMSD here gives you an idea of the precision of the final solution. Now, of course, we wondered whether this was actually correct, because it was the first time we did it this way. And luckily, we're collaborating with a guest in our institute, Masaki Nujiri, and he said, well, maybe I can try and crystallize it. We said, okay, you can try. People have tried for many years already, but he was not deterred by that, and in fact called us when we just had solved our NMR structure and showed us the crystal structure, which was rather surprising. And luckily, of course, well, mostly for us, because crystal structures have proven their value already for many years, the overlay was quite good, and that means that in solution and in the crystal, essentially they take the same position, and the putidaridoxin binds in the same orientation. So indeed, the paramagnetic method works well, provided that you take all these different types of restraints into account, because that was clear. If you leave one out, you will get a poor solution. Okay, having said that, I would just want to finish with two examples of work that we've done recently on new probes. One was for EPR, and just to say, for those of you who don't know, in EPR you have this method called DEER or PELDER, where you have two probes on a protein, and you can very accurately determine the distance between these two unpaired spins here with EPR, and you get these kind of modulations of your signal, and in that modulation is hidden a distance of the dipolar interaction between the spins, and you can derive that distance with some kind of distribution with uncertainty. And we were wondering whether we can make a probe from our clamps to do this with gadolinium. So we took the clamp 5 that I mentioned before and just changed the arm to make it into a probe that forms a thioether rather than a disulfide bridge. So we could also use this in cells for DEER experiments, because there the environment is very reducing and the disulfide bridges would reduce, so you would lose the probe immediately from the protein. And here the chemical work was done by Jing Miao, and Enrico Zurlo did all the EPR work on both of them PhD students in this project. So we synthesized this, and you can see here we varied the length a bit of this linker here. So with 2, 3, or 4 carbons, so N was 1, 2, or 3 here, to see whether that would have an effect on the DEER. We first took the clamp 5 as a reference, and put it on T4 lysozyme as a test system. We measured the standard pseudo-contact shifts and used that to determine the metal. So we have 2 mutants of the T4 lysozyme, different spectra. All that worked fine. Now we get a nice correlation between the calculated and measured PCS here for both of the mutants. You get these nice representations of the paramagnetic effect, so negative pseudo-contact shift, positive pseudo-contact shifts. And you see here an isobar of 0.4 ppm. So and this is the T4 lysozyme. So with that, you can determine pretty well where the metal is relative to the protein using these pseudo-contact shifts. So we did that to determine where the metals are on the basis of NMR data. And here you see that. So we modeled these 2 probes on one T4 lysozyme, and then you can see the distance that we measure with NMR at least is 44 angstrom using clamp 5. And the clamp 13, which has these other arms, should be very similar, of course, because you bind it to the same cysteines. So then we did these experiments for these clamp 13 with gadolinium in there, with strong PRE agent. So that is good for EPR. And we had these different lengths here of the linkers, A, B, and C, gave very similar results. So here you see the DEAR traces. And after that conversion for the distance distributions, you get 44 to 45 angstrom. So a very good agreement with the NMR distances. So that was encouraging for us, and we thought that seems to work pretty well. And then we tried in cell. Now that was hard to do, mostly to get spin-labeled proteins into a cell, into a living cell. We used dictyostelium cells for that are well known for taking up proteins easily from outside. And we could show that they took up the protein, first using fluorescent proteins, and that they also stayed alive. And after the experiments were still able to grow. So they actually take up also the spin-labeled proteins, but you see the signal is really noisy because the concentrations in the cell are very, very low compared to the in vitro situation. So we can get a distance distribution. It's rather broad here because of it's quite noisy, so it's not as reliable as in vitro, but it still works, and you can get distances for even of the 2 spin labels bound to a protein inside the cell. So that is quite nice, a method, I think. Jing also went on to do another type of metal probes, and this was for transition metals. So far everything was with lanthanoids, and we wondered, can we also put in cobalt or manganese, for example, in these type of probes. So for that we designed this one, and we called them TRAMs instead of clamps for transition metal NMR probe. So TRAMs, again the cyclin ring here, and now carboxy groups on either side with an additional hydroxy group, which also makes this thing asymmetric here, and then the 2 arms to link to the protein. So this was the one that we were interested in, and we made these as control probes here. So you see that this one has 1 arm and this one has no carboxy groups. And this model compound was published before, and we synthesized it again to compare it because it binds to cobalt to see how it compares with our structures. And first we added the manganese to this probe and then attached it to the protein. And you can see, indeed, we see the PRE effects. So all the peaks remain in the same position, but some of them are completely broadened out by the strong PRE effects of the manganese. It's the blue and red again, as I'm showing you before. So indeed, there's strong PRE effects from manganese. And you can then measure, as a function of the residue, the change in the intensity, so the I-para intensity under the paramagnetic sample and in the diamagnetic sample. And we see this effect here. So this is 1, so that means that this part is far from the probe, and the ones where it's almost 0 means that they are very close to the probe. And of course, we know the distance. We know where the metal is already from the modeling and the lanthanoid work. So we can make this graph here of the calculated distance from the PREs, and from the calculated from the structure, and this from the experimental PREs. And you see this trend here that it is between 20 and about 31 angstrom or so. You get reliable distances with an error of about 3 angstrom. Above this, there is no PREs, so you have an upper limit here, and below this, the signal is broadened out completely, so you have a lower limit here of the distance on this side. And you can visualize that better in the protein itself. So here we have the TRAMP1 with the manganese inside. All the red area, all these nuclei will be gone. In the green area, we have reliable distances, and in the gray area, you don't get any PREs simply because it's too far away from the probe. And so in this way, you can see where you can measure useful distances with manganese, in this case, in T4 lysozyme. And then we put in some cobalt to see what happens there. So this RR refers to these asymmetric centers that we have. We have RR or SS, but they behave very, very similarly. And again, you get nice pseudo contact shift here with the cobalt, indicating also that the cobalt is behaving as a nice paramagnetic center. We used 2 different proteins here. This is T4 lysozyme. This is another enzyme that we have, BCX, silanase, and this mutation here only is there to make it an inactive silanase. We don't want it to start working or anything. We could have used the active form, but this is the one we use anyway in the lab usually. And these 2 cysteines are for attachment of the probe, of course. So both of them gave nice shifts. And here you can see the tensors attached to the T4 lysozyme and to this BCX protein. Again, you get these similar plots here of negative and positive pseudo-contact shifts. And here you see that modeled in, we determine then where the metal is going to be for the RR and the SS. It's essentially the same position, and also the tensor orientation is very, very similar for both of them. And you see that here for the other protein. So they are reproducible for both the SS and the RR form. But what we noticed is that the size of the tensor is different. It's different for different proteins, for the different forms here. In other words, the size is very sensitive, so the absolute size of the paramagnetic effect, to the exact conditions. The type of probe that we use, the type of protein. Here we also tried it on ubiquitin. There it's quite small, and here it's more than 2 times bigger. And that is different from lanthanoids, where you always have more or less the same size for the tensor. At least in our hands, for example, with ytterbium, we always get around 8 times 10 to the 30 minus 32. So we don't really understand why that is. Apparently, the coordination of the cobalt is very sensitive to its environment, and maybe other residues have an effect on it. So yes, this is showing that. And we also compared it with this reference compound that I said was published before. There it's even larger. That is 7 on this scale here. So there's a very strong cobalt, gives very strong cobalt paramagnetic effects. I notice here that we use a QA. This is always a big bit of discussion here that I have with referees. It's a reduced form of the standard Q because we divide by both the calculated and the experimental value. We believe that this is in fact a better measure for the quality than just the Q. If there's interest, I can in the discussion explain why we think that this is in fact better. Of course, that means that effectively these values for a normal Q would be twice as large. So anyway, here's a model of this TRAMP modeled onto the protein, and you can see here's the cobalt. Here is the 2 arms, the carboxy groups coordinating to the cobalt, and the nitrogens in the ring. And for example, when we modeled this one, this tyrosine was very close and could readily be brought into this in hydrogen bond distance here. And you could imagine that has an effect on the tensor size already, because it will pull away the carboxy slightly from the cobalt and maybe redistribute the unpaired electron slightly over the orbitals. So we think that these kind of weak interactions with surrounding residues may well have an effect on this tensor. And then finally, I just want to show you a comparison because we have the same T4 lysozyme mutants here for the TREMP and for the CLAMP. And then you can see that the two are oriented really in a different way here in the models. Here the ring is more or less perpendicular to this alpha helix, and here the ring is more in line with the helix, and the cobalt, the lanthanoid is underneath the ring here, and here it's on the side of the ring. And you can see that also in the tensor orientations for the TREMP and the CLAMP, they are really in different orientations, as you also see here. Of course, the cobalt gives weaker effects, so it's more for more localized effect. If you want to measure paramagnetic effects close to an active site, for example, then the cobalt would be better. If you really want long-range information, then I would suggest to use lanthanoids rather than transition metals. So they have different properties, and also therefore different applications would be more favorable for them. And that brings me to the end, and I would like to thank a few people. Already mentioned the people here in bold. Simon was also a PhD student in the lab, contributed much to the P450 project and also a bit to the probes later. Technicians who always help us make all these proteins, several students. And especially I want to thank Mark Overhand, who has been my long-standing collaborator with making all these probes. He's the chemist, I'm only a biologist, as Gerard already mentioned. And all these designs of all these different probes are from him, and he helps the PhD students in the chemistry lab. And Martina also have been working for a long time with on EPR, and she was of course heavily involved in the EPR work on the new probes for deer. And I'd like to thank Masaki for the crystal structure that I mentioned before, and his students, and Harald Swalbe for giving access to the facility there, and when we did all these measurements on the P450. Just an advertisement, we still have a position open for a new PhD student to continue this work on paramagnetic probes. So if you're listening and interested, write to me. You still might get a position there. And with that, I would like to thank all of you for your attention.
Gerard, will you handle the questions, which you questions and answers? Great.
Fine. Are there any questions or remarks?
They are the questions and answers, which you find at the bottom of the screen. There's a Q&A, and if you open that, then you find questions there.
All right.
At the moment.
Okay, so I have a question by Nikolai Klishin. Do you hear me?
Yes, I hear you. Yeah.
Okay, Nikolai, go ahead.
Now you can just read it out, I think.
Yeah.
They cannot access him.
Okay, shall I read it out then?
Yes.
Okay. How flexibility of the LBT affect the spectra? How can you make a conclusion that LBT is flexible?
Yeah, LBT is the lanthanide binding tag, I presume. So you can see that because there's averaging, especially of the pseudo contact shifts, right? So if the probe is moving relative to the protein, that means that also the tensor that determines the paramagnetic effect is taking multiple orientations relative to the nucleus, and therefore the paramagnetic effect generally will be lower. It is averaged out, just like you have with RDCs, for example. So you will get lower values, and that is very clear. We have, for example, the CLAMP1 with the CLAMP5 with one arm versus two arms. If we attach it only via one arm, then the paramagnetic effects are about 5 times lower or so. So the overall size is a clear indication of how mobile it is.
Okay, there's another question by Nikolai. What software do you use for PCS calculations? Did you hear the question?
Yes, I hear the question. Yeah, we use for the calculation. Well, we measure the PCS, of course, and then for the tensor calculations, we use NumBot, a program developed in the group of Otting in Australia. And for structure calculations, we used Explore to put the pseudo contact shifts or the PREs in the computer and dock the structures. We use Explore NIH.
Okay. Then there is a question by Walter Gazin. The cytochrome P450 putida redoxin complex you're looking only at signals that are not broadened out by iron in heme and iron-sulfur cluster. Is that correct? And how were the assignments made in these circumstances?
That is correct, yes. So the heme groups have, in fact, pretty good properties. So there's very little broadening by heme groups. We use iron 3 there, that is paramagnetic, but the electronic relaxation time is very fast. That means that you get very little broadening. You do get some shifts, pseudo-contact shifts around the heme, but that was not a problem. The iron-sulfur cluster, on the other hand, is broadening a lot. So there we lose a lot of assignments of nuclei around the iron-sulfur cluster, and we only use the nuclei that are far from the iron-sulfur cluster, far on the backside. That is why I also showed this 7 nanometer thing in the beginning. We only can use the ones that are on the backside to do the docking, because on the front side, close to the iron-sulfur cluster, we simply don't have any nuclei. So that's a good point. From the heme point of view, as I said, we can see almost all the backbone. This is all backbone work here. We could see almost all the backbone nitrogens and protons.
Right. And there is a question by Liliana Quintanar. When using your lanthanide probes between 2 cis residues, how far do these residues need to be in the protein structure? I think how close they have to be.
Yeah. So what works very well for us is the alpha helix. So if you have a cysteine on I and then on cysteine I plus 4 or I plus 3, so 1 turn above, that gives a very nice binding, usually, especially of the clamps in our experience. Depends on the other probes, may have other properties, of course. And on beta sheets, so when you have 2 beta sheets, you could have neighboring residues and also link them together. You don't want the cysteines too close together, because then they form a disulfide bridge, of course. So the C alphas 5-6 angstroms away from each other at minimum, up to 8 angstroms away maximum, I would say, for the alphas and the cysteines, C alphas.
Right, then there is a question by Garrett Eaton. He asked what temperature is that the measurements were made?
Good question. I think this was all room temperature, but I don't know exact value, whether it was 20 or 25 degrees or maybe 30. I would need to look that up. It's or the deer, it depends. Yeah, if it's the deer, that is of course low temperature. So the deer only works at cryogenic temperatures, because otherwise the electronic spins are relaxing too fast and you don't get any EPR signal.
All right, and there is comment by Frans Mulder.
Hi, Frans.
Frans Mulder, yeah. Very nice talk, Marcellus. Are the slope variations in PCS simply due to distance and or angle variabilities, or is there more to it?
The slope variations, I presume you refer to this picture of the spectra with the slope with 1, and that the variation there is because the proton and the nitrogen, or the proton and the carbon, are not exactly on the same position. All right, so you have the probe here, and then you have the proton and the carbon sitting here, and they have more or less the same chemical shift, the same pseudo contact shift, I should say, because they more or less have the same distance and the same angle to the tensor, but not exactly. And especially if they're close to the region where the magic angle, where you have a very sharp change from positive to negative, for example, it might well be that the proton and the carbon have not exactly the same pseudo contact shift, and then it will not be exactly on the slope of 1. I hope that explains it. It's
okay. Then there is a question,
comes back.
There's a question by Daniel Heisinger. He says it's very nice work, Marcellus. The difference in the anisotropy could be due to linker mobility, conformational changes on the cage, or interaction with additional donors from the protein. Which is it, in your opinion?
So then he refers to the cobalt changes, I think. Yeah, as I said, I think it has to do with the coordination, because it's exactly the same probe on different positions, but yeah, if it is more mobile cannot be excluded, I suppose, that it is a bit more mobile, but a factor of 2 difference or even more I mean, we were surprised because we don't see it with the ytterbium, and why would you see it only with the cobalt and not with the ytterbium? And we used exactly the same mutants for the ytterbium. And there it is, yeah, I mean, it can vary a little bit, of course, like 5 or 10% or so, but not more. So intuitively, I would say the coordination changes, but I'm not an inorganic chemist, so I don't know enough about cobalt chemistry to be really certain about this.
There is a comment from someone who says that he would like to answer this question. So can you step forward? It's MPI, yeah. MPI BPC webinar admin. The name is not given here. Would like to answer this question. So
perhaps you can raise a hand and then we can promote him or her.
Okay. Then I go on to Frans Mulder again. How about domain motion on the DIA data? T4L is known to have domain motions on the nanosecond to microsecond timescale. Do you see any evidence of it? Or does it affect your analysis in any way?
Yeah, Frans himself actually studied this domain motion, I remember, from T4 lifetime. Indeed, well, we didn't look into this, I have to say. We just need to do more work. And it could well be that we saw a distribution that was not as sharp as we had hoped for distance distribution. And it could well be that domain motion has a role in this, yes. It's a good question, and I don't have an answer yet, as yet.
Okay. Then there is an anonymous attendee. He asked which factor determines that PCS will be upfield or downfield?
It depends where the nucleus is relative to the tensor. So I've shown these shapes looked a bit like orbitals with red and with blue. And if the nucleus happens to be in the blue area, it will be a positive shift, and happens to be in the red area of that shape, it will be a negative shift. And as I said, it's an isobar, so you can also make it bigger and show it for 0.1 ppm or 0.2 ppm. So when it's in the red area, it's negative, and when it's in the blue area, it's positive. It depends on the orientation relative to the frame of the tensor.
Okay, then another question by Nikolai Klishin. Do you expect different tensor size for different lanthanides in the same position?
Yes, definitely, because different lanthanides have different paramagnetic properties. So there's this whole series with different paramagnetic properties. As I said, the gadolinium, for example, doesn't give any shifts because it's an isotropic distribution of the electrons. That only gives PREs. Isotropic also means slower electronic realization time, so strong PREs. And the different lanthanoids have different properties. Europium has very weak, ytterbium is kind of in the middle, then you have holmium, which is a bit stronger, and then you have dysprosium and thulium, which are very, very strong. And that has nothing to do with the protein or with the cage, just with the properties of the electrons in the lanthanoid.
Okay.
But it doesn't help much to use to put two different metals in the same position in the same probe. You'll just get bigger shifts, but they're just scaled by a certain factor that you can predict more or less, and it doesn't help you to get more structure information usually.
All right. There's another question by Nikolai. Did you use peptide LBT? Is it better?
We didn't. As I said, there's many different things published. So definitely there's also lanthanoid binding peptides that are inserted into protein loops, for example, or the N-terminus or the C-terminus. And again, they have also their advantages and disadvantages. You disturb your protein a bit more usually, but they are relatively rigid, often relative to the protein. So the group of Harald Schwalbe started with that, if I'm remembering correctly. So you could look there and look for peptide lanthanoid binding tags.
Okay, then there is an anonymous question and comment. He or she says, very interesting talk, many thanks. Did you ever see expression problems due to the cysteines close in position?
Yes, so we always design several mutations, several positions for the probes. Of course, if you have cysteines already, especially on the surface, you would need to remove them from your protein first, because otherwise the probe doesn't know where to go. So if there's an internal cysteine, usually it's not a big problem, but if it's on the outside, you need to remove it. And then sometimes you see suddenly that your protein doesn't express very well anymore, or it aggregates, or it goes into inclusion bodies or so. So yeah, we definitely make several and then only pick the ones that work well. And I would recommend it to everybody. But as I said, if you take residues on good, nice, stable alpha helices that are clearly exposed to the solvent, that works pretty well usually.
Okay. The second question is, are the anisotropy tensors of all clamp generations predictable?
Of all clamp generations? Well, certainly not of the tramp, as I said, but this is clamp. Yeah, to a reasonable extent, I would say yes. I mean, because they have these two arms, you can predict pretty well where they're going to be within 3-4 angstroms or so. And as I said, for most of the lanthanoids, well, we have not tested all of them, but for the ytterbium and the holmium and the thulium that we use a lot, we can pretty well predict what it's going to be. I mean, yeah, not super accurately, of course. I mean, you still need to measure things, right?
Okay,
but, yeah.
That's clear. Then we have a question from Maria Angeles Canales. Which software do you use for back calculation of PRE?
So far, also mostly Explore NIH.
Okay, so there's a quick answer, right? Then there is a question by Manon Bruno. Can you detail why some metals give a strong PRE and low PCS, and with others it's just the other way around?
Oh, I can say briefly, this you can find in many reviews. It has to do with the electronic relaxation time. So you have this unpaired electron, and itself is also a spin that can relax fast or slowly, right? So if it relaxes slowly, then it will give you strong PRE effects. That has to do with how it relates to nuclear relaxation theory. So it needs to be in the nanosecond range or in the microsecond range. And then you generally get no PCS, because those are usually isotropic things. So like an MTSL, for example, has a slow electronic relaxation time, and that will give you no pseudocontact shifts, because there's very little anisotropy in the electron distribution. Same for gadolinium. When you have anisotropic electron distribution, then usually you have very fast electronic relaxation times, 10 to -12 or so, or 10 to -11. That will give you strong PCS because of the anisotropy of the electron distribution that you need to get pseudocontact shifts. But the PREs then are much lower. You will get more Curie effects, but yeah, and that goes too far, I think, to discuss here. But there's beautiful reviews. There's this book by Bertini, Ivano Bertini, and his colleagues about the theory of paramagnetic NMR. So please, I refer you to that.
Okay, thank you. And then there is Grace Royapa. Are there any paramagnetic probes that can cause less line broadening? Is there any software that can suggest positions in the proteins for incorporating these probes safely?
Less line broadening, I mean, well, you cannot have less. Not quite sure what you mean with less. I mean, you cannot sharpen up your peaks in principle, and except with some kinds of interference, relaxation interference. But normally, as I said, if you take the lanthanoids that have strong, fast electronic relaxation times, you will get little line broadening relatively, and good pseudocontact shifts. So you need the ytterbiums, the thuliums, the holmiums. But of course, if you have very strong pseudocontact shifts, you will also have more PREs. So you choose your metal wisely. If you want to measure something really far out, then you take strong paramagnetic centers. If you want to measure something relatively close to the center, you take cobalt, you take nickel, or you take europium or something. And is there software to predict the positions? Not that I'm aware of. I know that people have worked on this. I'm not sure it has been published to put these probes somewhere safely.
These will be usually be at the surface, right?
So we, as I said, we put them on the surface. I gave some indications of the distances, and we usually try out several things. That's because I'm an experimentalist, and I believe modeling can only get you that far. And there's a few rules of thumb that we also have published, like I said, clearly on the surface, ideally not charged residues, the distances that I mentioned, 5 to 8 angstrom away from each other or so. And then just make a few and try out.
Okay, thank you. Then there is the last question by Nikolai again. Does PRE also become less if the LBT is flexible or only PCS?
No, that is unfortunate. It will just become less reliable because the PRE will, if the probe is moving around, then it means that your distance is less reliable because it is not determined by one position of the probe relative to the nucleus, but by many positions of the probe relative to the nucleus. So again, you get a degree of averaging, but it not necessarily becomes much weaker. And that has to do with the R to the 6 dependence, R to minus 6 dependence. So if it's a bit close, you will get a very strong effect. If it's a bit further, it drops down very quickly. So the PRE works really in a different way than the PCS in this respect.
All right, thank you very much.